to my coronavirus classroom. I'm Janessa Jacobs, and this is blood. So blood is important for transporting stuff all around the body. That's probably the most obvious of its functions. Whatever you've got to get from one place to another is going there in blood. So transport is pretty obvious, but what is it that we're transporting? Well, we're transporting our respiratory gases, oxygen to the tissues, CO2 to the lungs. We're transporting our nutrients from the small intestine to the body tissues, and then our waste to the kidneys to be filtered out. Liver filters out waste as well. We're transporting things like hormones from our endocrine glands. So really, transport is a big, huge, important function of blood. Our respiratory gases. So O2 from the lungs to the tissues, and CO2 from the tissues to the lungs. That's happening in blood. We're transporting our nutrients and our wastes. We're transporting hormones. And that's really what you're transporting like naturally. But if you take drugs, that's being transported around in your blood. Alcohol, that's being transported around in your blood. Anything that you put into your body and it's got to get from one place to another is being transported around in your blood. So that's probably the most obvious function. The next functions aren't necessarily so obvious. The blood also helps with regulation. And what does the blood help regulate? Well, it helps regulate body temperature. If you recall, the first example of negative feedback that you learned about was body temperature regulation. And the blood has a huge role in that because blood is hot. If you recall from Gen Bio, cellular respiration produces heat as a byproduct. And your blood carries all of that heat to different places around the body, and that's what gives you your body temperature. So if you're super metabolically active, then you're going to get really hot and come above 98.6 degrees, right? Well, your blood vessels are going to dilate and take that hot blood to the surface of your skin, where we have sweat glands that we can turn on, and we can lose heat through evaporative cooling. And the inverse is also true. If you're too cold, we can take this warm blood from the periphery, and we can pull that warm blood into the vital organs that have to be maintained at body temperature. So blood is going to help with the regulation of body temperature. So we'll say blood carries heat and can shunt it around the body. I'm going to let you recall how, again, when it's cold, it'll shunt it into vital organs. When it's hot, it'll shunt it to the periphery so we can sweat. And this blood heat through evaporative cooling with those sweat glands. Okay, what else does the blood regulate? It actually helps regulate pH. So the pH of your blood has to be maintained between 7.35 and 7.45 on the pH scale. So it's slightly alkaline. And there are buffers and proteins and things in the blood that can make sure that if there's some change in hydrogen ion concentration, we can still stay in that range. So we say that it helps with the regulation of pH. And what we can say about that is that proteins and like bicarbonate ions, so proteins and ions in the blood are going to protect against changes in pH or rapid changes in hydrogen ion concentration. Okay, and then the other thing that blood is going to help regulate is fluid balance. So, or the movement of fluid between the intracellular and the extracellular compartment. It really helps what we say like push osmolarity. So the liver does about 200 things for the body, but one of them is to produce a bunch of plasma proteins. Some of them are called albumins, and those thicken up the blood. And so that can help to increase the viscosity if, say, we were hypotonic before. So basically, another thing that blood is going to help regulate is fluid balance, and that will be by guiding osmotic pressure to move into and out of the cells. So we'll say that plasma proteins contribute to osmolarity. And help guide the movement of water into and out of the intra and extracellular fluid compartments. There's an entire chapter of your book dedicated to that that you can read over later, to fluid balance. Okay, what else? The blood is also going to help function in protection. And what is it protecting us from? Well, our white blood cells are protecting us from disease and pathogens, so that's one thing. But blood also protects us from blood loss. And that's because it contains the plasma proteins that are going to be important in forming a blood clot if you nick a blood vessel. So for protection, we can say that white blood cells, which I will abbreviate as WBCs, white blood cells, protect against disease or protect against pathogens. And so that's one thing. And then blood actually also protects against blood loss. We get some protection against blood loss because there are plasma proteins that are pulled from blood to help form a blood clot. So our protection, we could say blood prevents blood loss, because our clotting factors are floating around in it. So platelets and then these proteins that are actually going to form the clot. So we are protected against blood loss due to clotting factors. And we'll talk about this in just a little bit. Alright, so transport, regulation, and protection. I just have a quick question for you then. Which of the following functions is associated with blood? A, delivery of oxygen to tissue cells. B, transport of digestive enzymes to the intestine. C, maintenance of DNA to convert egg cells. D, transport of neurotransmitters across synapses. E, electrical stimulation of the heart. Which of the following functions is associated with the blood? The answer is A, delivery of oxygen to the tissues. Okay, blood. What's it made of? Well, to figure out what blood is made of, what we do is we take whole blood from a patient, and then we put it in a tube, and we put it in this machine called a centrifuge, and it spins it around really, really fast, and all the heaviest stuff falls to the bottom of the tube. And that's how we can tell what it's made of. So if you were to look into a test tube of blood, what you would see is that the bottom, about 45%, is red blood cells, which are called erythrocytes. And then less than 1% of blood is this little teeny tiny layer right here called the buffy coat. And it's about 1% or less. And it's called the buffy coat. And what the buffy coat has is our white blood cells and our platelets. And the reason it's so low, such a low percentage of our blood, is that our white blood cells really don't belong in our blood. Their job is to fight infection, and infection happens in tissues. So white blood cells should not be high in the blood. They should be high in our secondary lymphatic structures. So our buffy coat's pretty small. Platelets are very tiny, tiny little particles. We'll talk more about them in a little bit. But then sitting all on top of this, then, the remaining about 55% of blood is plasma. So what is blood composed of? Plasma and formed elements. The plasma is this straw-colored fluid that you would find in the top of the test tube. We'll talk about everything that's in that in a moment. Our formed elements are everything else. They're the stuff that's formed. So our white blood cells and our platelets and our red blood cells. Our red blood cells are the most abundant formed element. And then our white blood cells and platelets are hanging out here in the buffy coat. Plasma makes up the biggest part of blood. Okay, so what's in plasma? Well, plasma is mostly water, so there's a bunch of water. And then everything in whole blood, like floating around in us right now, is whole blood. and everything that we find in blood is suspended in the plasma. If we pull it all apart and leave our cells and our platelets down there in the bottom of the tube, everything else that we find in plasma is like what? What would you expect? Like hormones, or your nutrients, or some free respiratory gases, very low levels free floating. 2% of your oxygen is floating in the plasma, maybe about 10% of CO2 is floating free in the plasma, but you got like bicarbonate ion, that's how most CO2 is transported. This is where your nutrients and gases are going to be transported around in. And drugs, medicine, that ibuprofen you took, your vitamins that you take, all of that is floating around in the plasma. So our plasma is mostly water, and then all of its dissolved solutes. So what are these dissolved solutes? We have like our ions, our vitamins, minerals, nutrients, so like glucose, all that stuff. What else is floating around in here? If you've taken any drugs, that would be floating around in there. Very low levels of respiratory gases. So about 2% of the oxygen that you have is floating free, and about 10% of your CO2 is floating free. What else do we have? We have proteins. So our proteins floating around in our plasma would be things like, we've got what are called albumins and globulins and different types of proteins, fibrinogen. So what are the ones that are most important to us? Albumins. So albumins are the most abundant plasma protein, and these are formed by the liver. And these are the ones that are going to have the biggest impact on viscosity or thickness, so the biggest impact on osmotic pressure. So they're formed by the liver, and these, we'll say, have the greatest impact on osmolarity. Okay, so they really thicken up the blood. What else do we have? We have globulins. So our globulins are going to be like our transport proteins that transport our lipid-soluble hormones, or our antibodies that are produced by our plasma cells, which are activated B cells. So our globulins are going to be produced by different cells depending on which type we're talking about. We're not going to go through all the nitty-gritty details of it, but what we'll say is that these can be transport proteins, they can be antibodies. There's a globulin that's part of hemoglobin and the red blood cells. We'll see that in a little bit. Another protein that's important, really the only other one that I want to focus on is called fibrinogen. Fibrinogen. Anytime you see an O-gen, it's going to make whatever we see in front of it. So what are we O-generating? Fibrin. And when it gets pulled from the blood, it gets converted to the insoluble fibrin, and that's what's going to clot and really prevent blood loss. So fibrinogen is soluble in blood, and it becomes the insoluble fibrin in a clot. And we'll talk more about that in just a little bit. What else do we find floating around in plasma? Non-protein nitrogenous substances? I don't know, those are all of our wastes. So wastes, ammonia, urea, uric acid, lactic acid, whatever is not a formed element, so a red blood cell, a white blood cell, or a platelet, anything else in your blood is floating around the plasma. So this antibodies thing is going to matter because we're going to talk about antibodies that you'll have in your blood to different blood antigens in a little bit. Okay. So what are our formed elements? This is everything that's solid and floats down to the bottom of your tube if you're centrifuging blood. And the most abundant of our formed elements are our erythrocytes, And as far as their structure goes, their structure is unique for a couple reasons. Now, the big function of erythrocytes is to transport oxygen. If you are going to be an oxygen transporter, do you want to use oxygen? No, if you're going to transport oxygen around, you don't want to use it. So, because then you're just like using up your cargo, and that's no good. So what happens in a red blood cell is that before it's ejected from the red bone marrow, it actually gets rid of its nucleus and its organelles, most of its organelles, and just like ejects all of that stuff so that it doesn't need any oxygen. So as far as its structure goes, what happens is during its development, it is going to eject its nucleus and organelles, and what's going to happen is it's going to become this shape that we call a biconcave disc. And that is because it ejects its nucleus and organelles right before it leaves the bone marrow. Okay, another thing about its structure is that its job is to accumulate a whole bunch of this stuff called hemoglobin. And hemoglobin is going to bind oxygen. So, before it does all this ejection of its nucleus and organelles, when it's developing through erythropoiesis, it's accumulating a lot of hemoglobin. I think it's like 250 million hemoglobin molecules per red blood cell. Each hemoglobin molecule can bind 4 oxygens. So each red blood cell has such a huge oxygen carrying capacity, it's quite magnificent. So structure. What we could say is that it's got a high amount of hemoglobin. And if you look at your book, what you'll see is that hemoglobin has these like globulin proteins that are all stuck together. The globin part is all these like big globby proteins. So each of these blue things is an iron, and that we'll say is the iron in our heme, or we'll say each of the blue part's a heme, and it contains iron. So you've maybe heard of anemia, people who are low in iron. If you're low in iron, then you can't make heme. If you can't make heme, you have a really hard time making hemoglobin, and then you have a hard time transporting oxygen. So there's iron in heme, and the heme part is what binds oxygen. So this is one hemoglobin molecule, which means that each hemoglobin molecule can bind 1, 2, 3, 4 oxygens. Okay, so that's kind of the structure. We accumulate a lot of hemoglobin. The heme part binds O2. And then the globin part is going to be able to bind CO2. Now, CO2 is primarily transported as an ion, but about 20% of it is bound to hemoglobin and our deoxyhemoglobin. About 20% is going to bind to the globin portion. So just be aware of that as far as the structure goes. Again, it's a biconcave disc, and we've just got a lot of this hemoglobin. Each of these hemoglobins is this crazy protein with iron in it. The heme binds our oxygen, globin binds CO2. That's what they look like. What do? They transport oxygen around. So the big function of an erythrocyte is to transport oxygen to the tissues. So function, transport oxygen to the tissues. You could say, yeah, but it also transports CO2, so isn't that a function? Sure, you could say it functions to transport respiratory gases. Great. But really, CO2 can transport on its own as bicarbonate ion. What we really need is oxygen to get to those tissues, and our red blood cells are doing that. So that's their big job, is to transport oxygen to the tissues. All right, well, how do we get our red blood cells? Well, all of blood is produced through a process called hematopoiesis, or hemopoiesis. And this is just our formation of blood. And that's a three-part process. We've got erythropoiesis, which is the formation of our erythrocytes, leukopoiesis, which is the formation of our leukocytes, and thrombopoiesis, which is the formation of our platelets. So the production of our red blood cells is called erythropoiesis. And it is stimulated in response to a drop in oxygen carrying capacity or low oxygen availability in the tissues, low red blood cell count, something like that. If the function of red blood cells is to transport oxygen, then what is going to get us to be produced is if we're not transporting enough oxygen. So the production is called erythropoiesis, and it's due to this hormone called erythropoietin. So if you've got a drop in oxygen or red blood cell or oxygen carrying capacity, something like that, then the kidneys are going to release this hormone called erythropoietin, which is abbreviated EPO, erythropoietin, and this is going to target the site of red blood cell production. What's the site of all blood cell production? Red bone marrow. So erythropoietin targets red bone marrow, and it's going to stimulate these cells. They are called hematopoietic stem cells. I'm not going to test you on that, but they're hematopoietic stem cells. It's going to stimulate them to go down the erythrocyte line. You do not need to worry about each stage of like proerythroblast and blah, blah, blah. I just want you to know the basics. That now in the red bone marrow, we're going to differentiate down the red blood cell line or the erythrocyte line. And what's going to happen, so we'll say our hematopoietic stem cell is going to develop down our erythrocyte line. Prior to ejection from the red bone marrow, it will get rid of all of its organelles and nucleus. Or it ejects its nucleus and organelles. And then it's going to leave the red bone marrow and enter the blood. So it leaves as a red blood cell or an erythrocyte and does its thing for about 120 days. So that is erythropoiesis. So, okay, I'm going to let Chip take you through a negative feedback loop that will get us some more red blood cells. Welcome to Negative Feedback with Chip by Janessa Jacobs. In this edition, we'll explore what happens when your blood oxygen drops. Hey good, what would happen if I were to take this pillow and hold it over your face? I think you might be in for a surprise when these wings put you in your place. Why? I was trying to think of the best way to get your blood oxygen to drop for this edition of Negative Feedback with Chip. Just try. I dare you. Forget about it. I was just trying to include you. When blood oxygen drops, this is detected by receptors in the kidneys, which will respond by releasing erythropoietin. Erythropoietin will target red bone marrow and stimulate erythropoiesis. The new red blood cells will be released to the blood, and oxygen carrying capacity will go up and homeostasis will be restored. So, erythropoiesis takes a little bit. If we need to immediately increase our oxygen availability, then another thing that erythropoietin can do is stimulate the spleen to eject all of those red blood cells it's been storing. Then all of the red blood cells that are now being produced in the red bone marrow can go and replenish that store in the spleen. So just be aware of that. Okay, what about the life cycle of a red blood cell? All right, well, it begins with erythropoiesis. So one, we have erythropoiesis, which again is if we have a drop in red blood cell number or a drop in oxygen availability. stimulate the red bone marrow to produce erythrocytes. Okay, so they go out and they circulate in the blood doing their thing for about 120 days. What's their thing? Transporting oxygen. So that's really the next big part of their life cycle, is for about 120 days, they circulate, transporting oxygen to the tissues. Okay, what happens after 120 days? Well, these cells are kind of dead for all intents and purposes. They don't have a nucleus or organelles. So what happens after 120 days is they start getting really rigid. Capillaries are the site of nutrient and gas exchange, and those blood vessels are super small. So small that the only thing that can get through them is one red blood cell, and it's got to bend to do so. And if you can't bend, then you're no good. If you're too rigid, you're no good. So after 120 days, I'm circulating around, I'm a red blood cell, I get to the spleen and there are test capillaries there. Also in the liver and red bone marrow to a lower degree, but the spleen is the big site of this. So in the spleen, blood's going to be coming through, and there are test capillaries that test for rigidity. And if I'm rigid and I can't get through, then macrophages right there hanging out in the white pulp are going to be able to gobble up that rigid erythrocyte and get rid of it because it's no longer useful to the system. So what am I going to do with all the parts? That's kind of like the fate of an erythrocyte at the end of its life cycle. So what we can say is that it circulates for about 120 days, transporting oxygen to the tissues. It will become rigid and is tested in the spleen. If it's too rigid, then macrophages there are going to gobble it up. So our macrophages will digest it and release the products. And release the parts. Well, what are the parts? Well, we had our hemoglobin that had heme and globin. Heme has iron in it. Iron is really valuable. So the iron is going to be recycled. All of the rest of heme is actually going to be converted to this stuff called bilirubin. Bilirubin is going to go to the liver. The liver is going to add that to bile. Bile is going to get concentrated in the gallbladder. You're digesting some meal. That bile is going to be squeezed into the small intestine. And now all of the rest of that bilirubin is going to go through the digestive tract and be excreted in feces. Bilirubin is actually what gives feces its color. So that's a little odd, but that's what's going to happen to the rest of heme that's not iron. What about the globin? The globin was protein. We have proteins everywhere. So if we can break down that protein, we can recycle those amino acids and use them somewhere else. So that's what happens. Our macrophages are going to digest our red blood cell and release its parts. What are its parts? From the heme, we'll say the iron is recycled. The rest becomes bilirubin and is excreted in feces. And the globin is broken down into amino acids, and our amino acids are recycled. All right, that is the life cycle of a red blood cell. Function of them now, I'm going to test you on the function of them now, and then we're going to talk about them again in the immune system, and I'm going to test you on them again then. Why? This is a comprehensive class, baby. Okay, so leukocytes, these are our white blood cells. They are going to protect us from disease. Alright, and we break them down into two groups. Granulocytes that have these really specifically staining granules, and agranulocytes that don't. We've got three types of granulocytes that we need to worry about. The first are called neutrophils. Neutrophils are the most abundant white blood cell floating around, so they tend to be the first to arrive at the site of tissue damage. And these are antibacterial. Neutrophils are antibacterial. They are the most abundant in the blood. And what are they going to do? They are going to be phagocytic for bacteria, meaning that it could gobble a bacteria up, but if a neutrophil does phagocytosis for a bacteria, it's going to die. It's going to eat it up and die. So that's okay, it works, but it's not a great way to protect the body. So instead what neutrophils can do, if there are a lot of bacteria, is something called respiratory burst. And if you recall from Gen Bio, all cells have these things called lysosomes that contain digestive enzymes. So what can happen in a neutrophil that senses there's a lot of bacteria around is it can take all of those lysosomes up to its surface and allow them to release their digestive enzymes. That's a process called degranulation, and what it's going to do is create this antibacterial kill zone with all of those enzymes. That's a process called respiratory burst. So they can be phagocytic, sure, but that's not great. It would be better to do this thing called respiratory burst. And what happens here is that the neutrophil will put its lysosomes to the surface of its cell or up to the plasma membrane, and they degranulate, meaning that they're going to release all of their digestive enzymes. This will create an antibacterial kill zone. Alright. Neutrophils are the easiest to see for a couple reasons. They're the most abundant, and they have these really characteristic multi-lobed nuclei that tend to stain really dark purple. So on the practical, I'll make sure that the lab assistant finds a good one for you to look at. So there will be a blood video with your lab assistant, so make sure you watch that and she'll show you what to look for. So if we were to look, a neutrophil might have a nucleus that's got all of these kind of crazy blobs coming all around. I mean, they're really, really distinct, super easy to identify. Okay, next, eosinophils. Eosinophils are the cells that are responsible for fighting parasitic worms. And for parasitic worms, they release neurotoxins that paralyze them. Eosinophils can also clean up after a specific immune response or an allergic response by gobbling up antibody-antigen complexes. So their big job is going to be to fight parasitic worms. And the reason that we call them, never mind. So they fight parasitic worms. Another thing they do is they ingest and process antigen, allergen, or antigen-antibody complexes. So after an allergic response, they can clean up, or after a specific immune response, they can help to clean up. And eosinophils, if we were thinking about what they look like, they have, to me they look like a little bit like cartoon barbells or dumbbells. They've got a bilobe nucleus, so it'll be kind of pink, and then the granules are kind of light pink. If I'm going to test you on it, I'll look for one that's got this nice, easily recognized kind of thing in the middle so that it looks like a barbell. Okay, basophils. Basophils are distinct in their indistinction. They pick up basic dyes, and they also have bilobe nuclei, but you can't see them because they're just kind of covered up in all these granules. So basophils, I think, are distinct in their indistinction. And basophils these are pro-inflammatory cells, so they release inflammatory chemicals, histamine and heparin. Histamine is histamine is a vasodilator, and heparin is an So these are the ones that are involved in allergic responses. Allergies like gone awry, you take an antihistamine to block down or stop all of this vasodilation in your nose. Alright, so those were our granulocytes, and again they get that name because they have specifically staining granules. Neutrophils are staining with neutral dyes, eosinophils are staining with eosin, basophils are staining with basic dyes. Our agranulocytes, you can still see cytoplasmic granules, but they're just like not in response to a specific type of dye. Our lymphocytes are about the same size as a red blood cell. And our lymphocytes are going to be involved in specific immunity. So we have two types. Our T lymphocytes, which are involved in cell-mediated immunity. And our B lymphocytes, or B cells, which are involved in humoral immunity. So we'll talk more about both of these when we get to the immune system. There's also a non-specific type of lymphocyte called a natural killer cell. So our natural killer cells are our anti-cancer cells. So those are all lymphocytes. Lymphocytes are about the same size as a red blood cell, and their nucleus is really dark purple staining and fills up almost the entire cytoplasm. So you'll see that it fills most of the slide, and then they tend to have like a little dip in them. So I'll make sure that the lab assistant points that out when taking you through lab. Our last type of white blood cell is the macrophage. This is also an agranulocyte. These are huge, so that's kind of the way that you'll recognize them. Our macrophages are our big eaters, so they're phagocytes, big eater. So they will ingest anything foreign, or they're going to try. So their big job is to be phagocytic, and they're going to try and eat anything that's not you. And they also become antigen-presenting cells. And we'll talk more about that when we get to the immune system. So macrophages, they're really huge. They've got kidney bean-shaped nuclei. And you can look at them more in lab. What about the production and lifespan of our leukocytes? Well, leukocytes are interesting because it's kind of going to be dependent on infection and what you're fighting. So all of them are produced by a process called leukopoiesis, which begins in the which occurs in the red bone marrow. So production is leukopoiesis in my red bone marrow. The lifespan is going to vary. If you're a neutrophil who just ate a bacteria, you're dead. If you are a memory cell to, let's say, tetanus toxin, then you only have to be boosted, what, every 10, 15, 20 years? You can live a long time as a memory cell. So I'm not going to test you on the nitty-gritty of that. You can take all of this in an immunology class someday. We're trying to get through blood. Okay, quick question for you. A white blood cell engulfing a bacteria is an example of blood functioning in A, protection, B, communication, C, regulation, D, transportation. A white blood cell engulfing a bacterium is an example of blood functioning in protection. That's right. And you might be looking at this and saying, well, communication isn't even a function of blood. Well, I would say, transport is, and if what I'm transporting is a hormone, then it might help with communication. Good things to think about. Okay. Platelets. Platelets are little cell fragments. They're formed through a process called thrombopoiesis. And what happens in thrombopoiesis is that hematopoietic stem cell up there in red bone marrow is going to become this big cell called a megakaryocyte. And this is going to stay in my red bone marrow. Well, my red bone marrow has lots of blood vessels around it. It must. If is stick its little feet out into this blood vessel, and then as blood is flowing by, it can shear off these platelets. So the blood flowing by pulls off platelets, and that's what they are. They are cell fragments, and they are, for just being cell fragments, they're actually pretty important. They release chemicals in platelet plug formation that are going to help attract more platelets to the area of bleeding if we're trying to stop bleeding. They can release factors that are involved in helping with tissue repair. So even though they're just these like little tiny cell fragments, they're really important. So we'll talk more about their function when we go through the specifics of hemostasis. Which is right now. So if you need to grab a drink, do it. So hemostasis is the stoppage of bleeding. And this word, hemostasis, looks a lot like homeostasis, which kind of looks like hematopoiesis, hemopoiesis. They all look similar, so just make sure when you're looking at test questions, you're looking at the word and answering the question being asked. Hemostasis is the stoppage of bleeding. And it's a three-part process. So let's just imagine we have this blood vessel here and it gets nicked. All of the blood vessels around it are going to be experiencing an inflammatory response, and we'll talk about that in the immune system. But this nicked blood vessel is going to be undergoing hemostasis. We have to stop bleeding. We need to limit blood loss. This is one of the protective things that's going to happen in blood. So the first thing that's going to happen, though, is our blood vessel is going to constrict to limit blood loss. So this damaged blood vessel has smooth muscle in its vessel wall. And the first step in this stopping the bleeding is for that muscle to constrict through this process called vascular spasm. So the first step in hemostasis is vascular spasm, and what's happening is that we have local constriction of the smooth muscle in the blood vessel to limit blood loss. So local constriction of smooth muscle in our damaged vessel to limit blood loss. Alright, so that's the first step. The second step in vascular spasm is called platelet plug formation. And in platelet plug formation, platelets are going to now, they usually are just floating by here and are repelled by the endothelium. So they're just usually normally floating by. But what happens in a damaged blood vessel is now for the first time we're exposing these platelets going by to the collagen fibers in the connective tissue out here. So what happens is the platelets are going to start sticking to this exposed collagen and secrete chemicals to attract more platelets that are going to stick to the exposed collagen to each other and secrete chemicals to attract more platelets in this positive feedback loop to create this platelet plug formation. So this platelet plug formation is actually one of the only examples of positive feedback in the body. It is the only example of positive feedback in males. The other three examples of positive feedback that we have happen in the female reproductive tract. So because of that, Good is a little upset. He does not get as much time as Chip does ever, and especially in this positive negative feedback thing. So I'm actually going to let him take over and describe this for you. So here's positive feedback with Good.
Welcome to Positive Feedback with Good by Janessa Jacobs. This is the platelet plug party, the only positive feedback party where males are invited. When blood vessels get damaged, collagen fibers in the connective tissue become exposed. Platelets will stick to the exposed collagen and secrete chemicals that attract more platelets. More platelets will arrive and stick to the collagen and the other platelets. They will secrete more chemicals to attract more platelets. When does it end? When the collagen fibers are all covered up. Positive feedback ends and coagulation begins.
Alright, so the platelet plug is going to temporarily stop bleeding. It's not a really strong plug, but it will kind of cause temporary blood loss. So for our platelet plug formation, what we'll write down about it is that it's this positive feedback loop that allows platelets form a temporary plug. So we'll say it's a positive feedback loop that creates a temporary plug. The most powerful step in hemostasis is coagulation. And what happens in coagulation, there are actually two methods and they're big cascades, and what ultimately is happening is we're trying to get prothrombin activated to thrombin, and we're trying to pull that fibrinogen out of the blood and into this tissue and convert it to the insoluble fibrin. Fibrin is going to form the clot, and it's going to grab all those platelets in the area and red blood cells in the area and all of the tissues that are damaged in the area, grab it and pull it together so that we can form a clot. So coagulation, this is what actually stops bleeding. This is our most powerful step. It stops bleeding. And what we could say is happening here is that fibrinogen is getting pulled from the blood and becoming the insoluble fibrin to form the clot. So fibrinogen pulled from blood and becomes fibrin. What fibrin is going to do is form a fibrous clot as it pulls our platelets and cells together. So at the end of coagulation, we have formed a clot and we have stopped bleeding. So if all you were asked to do on a test was to list the steps in hemostasis and briefly describe them, the steps in hemostasis are what? Vascular spasm, which is localized constriction of smooth muscle in the damaged blood vessel to limit blood loss. Platelet plug formation, which is a positive feedback loop in which platelet plugs stick to collagen, secrete chemicals that attract more platelets that stick to the collagen in each other, secrete platelets that attract more platelets, and so on until we form this temporary plug to limit blood loss. And then coagulation, in which fibrinogen is pulled from the blood to become the insoluble fibrin. That, now, we've stopped blood loss, right? That's hemostasis. We've stopped bleeding. All right, well, there are some other things that can happen. We can stabilize that clot, and then after it's no longer useful, we're going to break it down. But before we get to those steps, I have a quick question for you. So, platelets stick to A, blood, B, collagen fibers, C, fibrinogen, D, all of the above. All right, the answer is collagen fibers. Good job. All right, so clot retraction. After about 30 to 60 minutes, the fibrin is going to contract and pull together. So it's this fibrous kind of protein. So if we can pull together, then what we can do is we can pull that damaged tissue together, get the cells closer to each other, and kind of stabilize it so we can set the stage for tissue repair. So growth factors there can start really helping to get our cells back together and all that jazz. So in clot retraction, what happens is after 30 to 60 minutes, the fibrin contracts or retracts. It's going to pull our tissue together. It's going to stabilize the clot. And it's going to help set the stage for tissue repair. Okay. That is clot retraction. Fibrinolysis. Now anytime you see something's going to happen. Oh, now we're going to lyse that fibrin. How do we break down the clot when it's no longer useful? That's what fibrinolysis is. So after a couple days, when the clot is no longer useful, there are enzymes that the platelets actually release and that the healing tissue is going to release that's going to break down that fibrin so that the clot can be, just dissipate. And our macrophages will gobble up all the tissue debris and all that jazz, and then we won't have a clot anymore. So when the clot is no longer useful, we could say enzymes from our platelets and healing cells break down the fibrin. Okay, the next thing we're going to talk about is blood typing. And so in order to get there, we should start with some definitions. So an antigen is something that is going to stimulate an immune response. And we have self-antigens that mark our cells as self. So they wouldn't stimulate an immune response in us, per se, unless it was autoimmune, but they would stimulate an immune response in someone else. An antibody is something that's produced and marks antigens for removal from the system. So antigens are substances that can elicit an immune response. Antigens, substances that can elicit an immune response. And then antibodies are these proteins that can bind to antigens and mark them for removal from the system. So antibody, it binds antigen and marks for removal from the system. Now, how does this relate to blood? Antigens. We're going to have self-antigens on the surface of our blood cells that are going to mark us as self, and that's also going to be the basis for blood typing. So antigens. Self-antigens on our erythrocytes are responsible for typing. Okay, and then antibody. Well, these are proteins produced by activated B cells that mark antigen for removal. So if I've got a antigen on the surface of my red blood cells, then floating around in my plasma, I have anti-B antibodies. So antibodies are proteins that mark antigens for removal from the system. And whatever blood type you are making the antibody to the other blood type. So antibodies, what these are is they are plasma proteins specific to antigens. Coronavirus class, distraction, break. Okay, so why do I tell you this? Well, because we're going to talk about blood typing. And why does this matter? Well, because if you give a blood transfusion, then we need to consider what blood type is the recipient, what do they have floating around in their blood, what is present on the surface of their blood cells, what's our donor blood got, all that stuff. So this is what's going to help us figure it all out. So blood transfusions are when you give blood from one person to another, and in whole blood transfusions, we really try not to do whole blood transfusions. Because in a whole blood transfusion, what you're giving, or what you're getting, is like all of the plasma and all of the formed elements. So whole blood contains plasma and formed elements. So in that plasma, we have all of the antibodies. Antibodies for proteins. When we were talking about what we found in plasma, we said proteins are there. There's a lot of them, and antibodies are some of them. Okay? blood cells. So what instead you get when you're donating blood, they'll separate the components, and what you're getting when you get a blood donation is packed red blood cells. So this is just the red blood cells. So we pull off the white blood cells, we pull off the platelets, we pull off the plasma, and it's just red blood cells. Okay, why does this matter? Well, because of blood typing and the human blood grouping, and just thinking about like what we find everywhere, and all the blood is going to kind of help you think about like donor, recipient, pairing, and all of that for the test. So, okay, our human blood grouping is this ABO grouping system that we use. And this is going to tell you whether you're type A, type B, or type O. So I'm actually going to come and let's just start here and look at our blood types. So anytime you're looking at a blood typing question, what you need to ask yourself is what is on the cell and what is free in the blood. So what is on the cell is our first question, and what is free in the blood? So what is on the cell and what is in blood? So what's going to be on the cell, antigen or antibody? Self-antigen. Okay, so what is on the cell? Antigen. Okay, so what could be floating free in the blood? Antibody. So antibody. Okay, so let's just think about this. You've probably heard, and I just told you a moment ago, we had ABO groups. So if you're type A, that means your blood cells have all these little type A antigens, which are just self-proteins all over them. So if you're type A, you've got type A antigen. What that means is floating around in your blood as an antibody is the anti-B antibody. So floating, what is in my blood, is the anti-B antibody. Okay, so then we've heard you have type B blood. So if you have type B blood, then what you have on the surface of your cells are these B antigens. So that's what makes you type B. So if you have B antigens, then what's floating around in your blood are anti-A antibodies. Okay. Well, blood typing is one of those things that displays codominance, meaning that you can display both A and B antigen. So in that case, you would be type AB. And on the surface of your cells, we would find A and B antigen. So if you have A and B antigen on the surface of your cells, what type of antibody would you have? None, because you can't have anti-A or you'd be fighting your A. You can't have anti-B or you'd be fighting your B. So what is in their blood? No antibodies. Well, that's not true. They've got antibodies to everything they're immune to. No anti-A or no anti-B. Okay. Now we've all heard of type O. And on type O, these people have neither A nor B antigens on the surface of their cells. So there's nothing on top of the cell. What type of antibody might they have floating around in their blood? They might have anti-A and they might have anti-B. Okay, so, all right. You may have heard that type O is the universal donor. Really, type O negative is the universal donor. But type O is the universal donor. And that's because there is nothing on the surface of their cells. So I can give them to A, and there's no B for their antibody, anti-B antibody to fight. I can give them to type B, and there's no A for their anti-A antibody to fight. I can give them to type AB, and there's nothing, well, they wouldn't be a problem anyway. They're the universal recipient. So, since they're not making any antibodies, they can take anything. And I can give my type O to type O, we're good. All right, so this is why the packed red blood cell thing matters though. Because if I of O to somebody with type A blood, then the donor's anti-A antibodies would cause some agglutination. So what I do is I take a type O person's blood and I spin it down and I pull out all their plasma and I take away those anti-A and anti-B antibodies and I just give packed red blood cells that are O blood cells to anybody, and there's nothing to fight, so there won't be an immune reaction. Okay? So some things that we could say about this. Who can A's take blood from? So because they have anti-B, who could they receive blood from? They could take it from A. Could they take it from B? No, because they've got anti-B. Could they take it from AB? No, because they've got anti-B. Could they take it from O? Yes, because everybody can. Okay, what about type B? Who could I take it from? Could I take it from type A? No, because I'm making anti-A. So if you give me type A blood, my anti-A is going to bind it up. Could I take it from type B? Yes, so I can take it from type B. Could I take it from type AB? No, because my anti-A is going to bind up all this A. Could I take it from O? Yes. All right, I'm type AB. Who could I take blood from? Could I take it from A? Yes. I'm not making any anti-A. Could I take it from B? Yes. I'm not making any anti-B. Could I take it from AB? Yes, that's a match. Could I take it from O? Yes. Perfect. All right, I'm type O. Who can I take blood from? Can I take it from A? No, I'm making anti-A. Can I take it from B? No, I'm making anti-B. Can I take it from AB? No, I'm making anti-A and anti-B. So who can I take it from? I can only take it from O. Okay, so that's kind of the first and most important part for blood typing. The next thing is the Rh factor. So the most important thing for tissue donation and all of that is the ABO factor. The next most important thing is actually these things called major histocompatibility complexes. But then after that, we have to consider this other group of self-antigens that some people have called the Rh factor. So if you've heard, oh, I'm A positive, what it is that you're positive for the Rh factor. So you have Rh proteins. So if I'm A positive, then that means on the surface of my cells, I have A antigens, but I also have these little Rh antigens. So they call it the Rh factor because it was discovered in Reese monkeys, but this would make this person is A positive. Let's say this person doesn't have Rh, so they are B negative. This person does have Rh, this person is AB positive. This person doesn't have Rh, so they are O negative. Okay, so if you are Rh positive, then you have Rh. Would you ever make anti-Rh antibodies if you're Rh positive? No, never. So if you're Rh positive, you're never making anti-Rh. If you're Rh negative, then you're not immediately making anti-Rh. So no anti-Rh until exposure. But after you're exposed to Rh, you will make anti-Rh. Which means from now on, if you receive a tissue or organ transplant from anybody who is Rh positive, your anti-Rh antibodies are going to fight it. So don't think about that the other way. If you're O negative and you get O positive blood, you don't all of a sudden become O positive. If you're O negative and you get O positive blood, you all of a sudden start making anti-Rh antibodies to become immune to the Rh antigen. So be aware of that. Why does this matter? It doesn't matter a whole lot in blood transfusions. It matters more in this case of what's called erythroblastosis fetalis, or something like that. And what happens there is that you get a blood type mismatch between a mother and the baby growing in her uterus. And so if you've got an Rh negative mother who gets pregnant with an Rh positive baby, at first it's not really going to be an issue because the placenta is going to give some protection. But when she has that Rh positive baby, it's going to cause issues. So let's look at this. Let's just say here's our mom, and she is O negative. So this was actually my mom. She was O negative. And she gets pregnant with an Rh positive fetus. Okay, the first time, not a problem because she's not already making anti-Rh. So she has this baby, and it's a violent bloody process, and there's mixing of the blood, and she gets exposed to the Rh antigen. She will start making anti-Rh antibodies. She's had the baby, it doesn't really matter, but now floating around in her blood is anti-Rh. She gets pregnant a second time with an Rh positive baby. Now all of this anti-Rh floating around in her blood is going to bind to the Rh positive cells on this baby and attack them. So attack all those red blood cells, and then what happens is that, Theseus, it's not your turn. It's not your turn. So then what happens is that anti-Rh is going to bind to all of those red blood cells, and those red blood cells will be destroyed. As a byproduct of that, then, we get our heme broken down, iron's recycled, the rest becomes bilirubin. That bilirubin actually collects in these babies, and they get jaundice, or they start to look orange. And it's because their mom is attacking their blood cells. So the baby may or may not be able to survive this. So, like in my mom's case, she got pregnant with my brother. He was O positive. She had him, and they gave her the shot. They can give you a shot called Rogam to inhibit this immune response. So they gave her a shot of Rogam. She got pregnant again with another Rh positive fetus. She had a miscarriage. That baby died. They gave her a second shot of Rogam. She got pregnant with me. I'm A positive. And so she had me. She was attacking me still. So, but she had me at work. So they gave her the Rogam.
Wow.
And then she got pregnant again. That baby must have also been Rh positive, and she had another miscarriage. So they gave her a second shot of Rogam. And then she got pregnant with my little brother. And I don't know what his blood type is. She was able to maintain him. But what was interesting is that it seemed like her anti-Rh response was so high that they had to suppress it twice before she could maintain an Rh positive pregnancy. Just kind of an interesting thing to be aware of. So blood typing. Why does all of this matter? Well, let's say you have an unknown blood sample. We can actually stick all of these different antibodies onto that unknown blood sample and figure out its blood type. So why? Because antibodies are going to clump these things together and agglutinate it, and so you'll see clumping together of these blood cells. So let's just imagine that we've got an unknown. I'll do 3 examples, because as you can see, we've got company, and this is what makes the coronavirus classroom increasingly ever harder. So on each of our blood samples, we're going to put on anti-A. On each of our blood samples, we're going to put on anti-B. And on each of our blood cells, we're going to put on anti-Rh. They're all unknown. So we've got unknown number 1, unknown number 2, and unknown number 3.
Mom?
Yes?
I didn't know your mom is so old.
My mom's dead, buddy. Can I finish this lecture?
Mom's so old.
Okay, so let's say I've got unknown sample 1. And so I've got this unknown blood sample, and I split it between these 3 trays. So each one of them has unknown 1 in it. And I stick on anti-A and I get no agglutination. And I stick on anti-B and I get agglutination. And I stick on anti-Rh and I get agglutination. What blood type do you think this is? So if I stick on anti-A and there's no agglutination, then this cell has no A on it. If I stick on anti-B and there is agglutination, then there is B on it. If I stick anti-Rh on it and there is agglutination, then there is Rh on it. This blood type is B positive. Okay, now what about this one? I split up unknown blood sample 2 between my 3 dishes, and I stick on anti-A, and I get agglutination. And I stick agglutination. What blood type am I? Well, if anti-A is binding, I have A. If anti-B is binding, I have B. If there's no anti-RH, or if there's no RH, then anti-RH can't bind to anything, I'm AB negative. Okay, my third and final unknown blood type. I split it up between my 3 trays. I stick on anti-A and there is no agglutination. I stick on anti-B and there is no agglutination. I stick on anti-RH and there is no agglutination. That means I am O negative. There's no A on my cells to bind, there's no B on my cells to bind, there's no RH factor on my cells to bind, and I am done with my blood lecture. Thank you for attending my coronavirus class.